Hello everyone, and welcome back to the Catalyst Learning Center's Biochemistry course. I'm Patrick De Castro, and I'll be a lecturer for today's topic. Now for our discussion, we'll be discussing about something very important, the N-terminus and the C-terminus of an amino acid. Now this may sound a bit technical at first, but by the end of this discussion, we'll be able to understand why these matter when it comes to understanding proteins. But before that, I'll be taking your attendance first. So please comment down in the chat box present for me to confirm your presence in today's synchronous meeting. Now let's go.
So at the end of the session, the learner should be able to define N-terminus and C-terminus in the context of protein structure, identify the key chemical groups present at the N-terminus and C-terminus, explain the significance of the N-terminus and C-terminus in protein synthesis, structure, and function, describe various post-translational modifications that can occur at the N-terminus and C-terminus in their applications, discuss the applications of understanding N-terminus and C-terminus in the fields like protein sequencing and purification, and lastly, demonstrate the application of the N and C-terminus in real-life setting.
To the start of the discussion, let me first ask you a question to get our brains thinking. What do you think is the basic building block of a protein? If you answered amino acids, you are correct. Good job. Now, here's a follow-up question. How do you think these amino acids connect, or what do you think connects them to form something so complex as a protein? If you answered peptide bonds, you're correct again. And if your answers weren't quite right, that's completely okay. We'll be going through everything step by step, so it will all make more sense as we continue. So just to summarize everything so far, a protein is essentially a long chain of amino acids linked by peptide bonds, and we call this chain a polypeptide. Now, here's something to reflect on. If a protein is made up of a long chain of amino acids, do you think that this chain contains a starting point and an end point, just like a string? And that's what leads us to today's main focus, identifying the starting point and the end point of a polypeptide chain, also known as the N-terminus and C-terminus, which plays a crucial role in determining or to determine the structure and function of a protein as a whole. So let's begin by taking a closer look at the first end of a polypeptide chain, also known as the N-terminus or the amino terminus. Now, the N stands for amino or nitrogen because this N marks the beginning of a polypeptide chain. Now, here's a question. What do you think characterizes an N-terminus? Good job! So the N-terminus is defined by a free amine group. Now, this group attaches to the very first amino acid in the chain. Now, in eukaryotic proteins, that first amino acid is most often methionine. This is due to the way protein synthesis starts. Now, to help you visualize better, let us take a closer look at this diagram.
What you're seeing here is a signal peptide, which is frequently located at the N-terminus of a protein. It plays an important role in guiding the protein to its correct destination within the cell. So the signal peptide is made up of three regions, namely the N region, which is usually positively charged, then the H region, which forms a hydrophobic core that typically falls into an alpha helix, and lastly, the C region, which contains the cleavage site, which may form a beta sheet.
Now that we've taken a closer look at the structure of a signal peptide at the N-terminus, let me ask you this. What do you think these targeting signals actually do in the N-terminus? Exactly. They direct proteins to specific locations within the cell, like the endoplasmic reticulum. Now let's dig deeper. Have any of you heard of the term N-myristilylation? If you haven't, just based on the word, what do you think myristilylation might imply? Wow, those are some great ideas. So, N-myristilylation is a type of a post-translational modification, meaning it happens after or often during protein synthesis. It also involves the addition of a myristoyl group, which is a fatty acid, to the N-terminal glycine protein. This influences protein localization and interactions. To get a clearer vision of how this occurs, let's take a closer look at this diagram together.
So this diagram shows how the myristoyl group is attached to the protein during or shortly after translation. First, a fatty acid called myristoyl-CoA is made. Next, the N-myristonylation can happen either during protein synthesis or after the protein is made. Then, the enzyme NMT or N-myristoyl transferase binds to the myristoyl-CoA and forms a complex. After that, the protein that needs to be modified attaches to this complex. Finally, NMP transfers the myristoyl group to the protein, creating a myristoylated protein and releasing CoA. The result is that the protein becomes anchored to a membrane, which is crucial for its function.
Now that we've explored the N-terminus, let's move on to the other end of the polypeptide chain, which is the C-terminus. Now, can anyone tell me what chemical group do you think defines the C-terminus? That's right. The C-terminus is characterized by a free carboxyl group, and it marks the end of the polypeptide chain. So with the word carboxyl, the C-terminus stands for carboxyl, just like the N-terminus. Now this N also carries important signals or undergo modifications that affect where the protein goes and how it behaves in the cell. One example is the presence of retention signals, which helps keep certain proteins inside specific compartments like the endoplasmic reticulum. Another important modification that can occur at or near the C-terminus is called prenylation. Based on the name, take a guess. What do you think prenylation might involve? Those are some very great guesses. So, prenylation involves the addition of hydrophobic prenyl groups to a protein. Now, these groups help anchor the protein to membranes, making the modification essential for the protein's proper localization and function. Now, let's take a look at this diagram.
This diagram shows how a well-known protein called Ras undergoes prenylation. Prenylation happens in three main steps, each carried out by a different enzyme. First, an enzyme called farnesyl transferase adds a farnesyl group, which is a type of lipid, to a cysteine near the protein's C-terminus. Next, a membrane-bound protease cuts off that last three amino acids from the protein's tail. Finally, another enzyme adds a methyl group to the modified cysteine, making the protein more hydrophobic so it can attach to the cell membrane.
Now that we've explored both the N-terminus and the C-terminus, can anyone mention a key similarity between the two? Yes, that's right. Both the N-terminus and the C-terminus mark the structural boundaries of a polypeptide chain. They also serve as a common site for post-translational modifications, making them important reference points when studying for sequencing proteins. So great job with the answer. Now, that we've mentioned their similarities, what do you say is a key difference in their primary roles? Wow, that's a very great observation. The N-terminus is where protein synthesis begins, and it often contains targeting signals that help direct the protein to its proper location. On the other hand, the C-terminus marks the end of the synthesis and is often involved in the functional interactions or retention signals. Chemically, the N-terminus has a free amine group, while the C-terminus has a free carboxyl group. Now, after all this discussion, let's take a step back and think outside the box. What do you think, or why do you think understanding the N and C terminus matters in biochemistry and biotechnology? Well, those are some excellent examples. Now, there are several practical applications where knowledge of protein termini is crucial. First, let's talk about polypeptide modifications. Knowing where the N and C termini are allows the scientists to modify proteins to enhance stability, increases activity, or add up detection tags. Now, these modifications can help track where a protein goes in a cell or how it behaves in experiments. Second, the termini plays a big role in protein sequencing. Identifying the amino acid sequence is fundamental for understanding a protein structure and for applications like drug development and quality control in biotech. Now, one common method for sequencing from the N-terminus is called the Edman degradation. Now, can anyone tell me what mass spectrometry might be used for in this context? Great job! Mass spectrometry is used for determining the sequence or molecular weight of proteins and peptides, and it can identify both N-terminal and C-terminal modifications. And finally, in protein purification, scientists often take advantage of the termini by adding affinity tags like this tags or either end at the protein. Now, these tags act as molecular hooks. So how do you think this helps with purification? Yes, that's right. His tags can bind to nickel columns, allowing the protein to be easily separated from a mixture. This makes purification faster and more efficient. So as you can see, even though the N-terminus and the C-terminus may seem like small, complex details, they are essential for protein's entire life cycle, from how it's made, to where it goes, to how we can apply and study it in real-world situations. Now, as we reach the end of our discussion, let's take a moment to go back to our learning outcomes and check whether we've achieved everything we set out to cover today. First, we aim to define the N-terminus and the C-terminus in the context of protein structure. We've discussed how these two ends mark the beginning and end of a polypeptide chain. Now, the N-terminus starting with a free amine group and the C-terminus ending with a free carboxyl group. Second, you were asked to identify the key chemical groups and found at each terminus, and you correctly pointed out that the N-terminus has an amine group and the C-terminus has a carboxyl group. Great job on that, everyone. Next, we explain the significance of both the termini in protein synthesis, structure, and function. Now, we saw how the N-terminus initiates synthesis and often carries targeting signals, while the C-terminus concludes synthesis and influences interactions or localization. We've described various post-translational modifications that can occur at the termini, such as the N-meristylation and the prenylation, and how these affect protein behavior, localization, and function. Then we explored real-world applications of this knowledge in biotechnology. You saw how understanding termini plays a role in protein sequencing, such as through Edman degradation or mass spectrometry, and in protein purification using affinity tags like these tags. And finally, we looked at how these concepts apply in real-life settings, from designing drugs and target-specific protein forms to engineering proteins for industrial and medical use. So with all that in mind, I hope that you now see that the N-terminus and the C-terminus, well, just the ends of a protein, are actually central to how proteins function. Hopefully, you'll be able to apply the knowledge you have gained today in either the school, your future profession, or real-world situations. Now, talking about applications, later on I'll be posting an assignment or an activity for you to work on. So this is a by-group activity wherein you will have to apply what you have learned today. So by that, you are tasked to create a diagram that showcases how the N-terminus starts and how it ends with the C-terminus. So the whole sequence. So instructions and rubrics will be posted later on for you to look at, and hopefully that you'll be able to submit on or before the deadline. So, thank you everyone for your attention today. So if you have any more questions, do not hesitate to send me a private message and I'll get back to you as soon as I can. So again, this is Dr. De Castro, your lecturer for today's topic, and thank you for watching another video of the Catalyst Learning Center's Biochemistry course. Good job and happy learning.